You can hear me? Okay, today is day 2 and the audience is very limited, but we should start. So as we mentioned, like this talk is about the emerging disease, and as you know, the re-emerging disease, there are a lot of viruses. We are talking viruses only. So they are different categories, right? BSL-1, BSL-2. So, some of you, like UG's students, they don't know what is biosafety level 1 and 2. So just watch this video, get an idea what is BSL-1 and BSL-4. Yeah, please play the video. It is better always to see the pictures and videos, get better understanding than.
Biosafety levels consist of safety measures designed to protect workers and the environment against potentially infectious microbes and agents. Let's take a closer look at each biosafety level and their accompanying.
The sound?
Follow basic microbiological practices such as basic personal protective equipment like gloves, lab coats, and eye protection splashes. Considering this, BSL-1 labs don't need specialized facilities. Contaminated before disposal, ensuring safety and compliance with regulations. Potentially infectious microbes and agents. Let's take a closer look at each biosafety level and their accompanying safety requirements. Biosafety level 1 is the first tier in the hierarchy. It applies to agents that don't typically cause disease in healthy humans. These labs handle well-cared. Neutralize agents like non-pathogenic BSL-1 labs follow basic microbiological practices such as proper hand washing and safe handling of sharps. In addition, it mandates basic personal protective equipment like gloves, lab coats, and eye protection that are used to prevent skin contact and protect against splashes. Considering this, BSL-1 labs don't need specialized facilities. Still, biological waste is decontaminated before disposal, ensuring safety and compliance with regulations. Biosafety Level 2, or BSL-2, is for agents that pose moderate hazards. These agents can cause human disease, but treatments are usually available. Given the nature of biological hazards handled, BSL-2 labs restrict access when work is conducted. Biohazard signs and procedures for spills or exposures are strictly enforced. Aside from PPE, biological safety cabinets are used when working on infectious aerosols to ensure isolation and protection. BSL-2 labs also have more facility requirements. Facilities need automatic doors to contain hazards in case of accidental spills, along with an eye wash station and autoclave nearby for decontamination. Biosafety Level 3, or BSL-3, involves handling microbes that can cause serious respiratory diseases.
These pathogens require special precautions due to airborne transmission risks.
See the suit they
Required
wear.
PPE include respirators, face masks, and gowns. In addition, all work is done on biological safety cabinets or enclosed equipment to prevent exposure. BSL-3 labs are physically separated and maintained at negative pressure. All exits are sealed and decontamination methods like autoclaves are available within the containment zone. Biosafety Level 4, or BSL-4, is the highest level for handling dangerous and exotic agents. These pathogens pose a high risk of aerosol-transmitted
They have air
infections
pressure.
and often lack treatments.
So the oxygen coming
Access to
into the
BSL-4 labs is strictly controlled. Only authorized, highly trained personnel can enter, and all procedures are conducted in Class 3 safety cabinets or while wearing positive pressure suits. As an additional safety measure, personnel wear full-body suits with a dedicated air supply. They must undergo a full-body decontamination process before entering and after exiting the lab. BSL-4 labs are isolated and completely sealed.
Every time you
The
go in and
facility
come
is
out.
designed to prevent leaks, with all materials exiting the lab being thoroughly decontaminated. Understanding biosafety levels is essential for maintaining safety in various laboratories. For more information about industrial safety, like and subscribe. Stay safe and see you on the next one.
Okay, back to the slide, please. So this is an idea like how BSL 1 and 4. So I think your institute is a lot of BSL 1. I don't know BSL 2 you have any hood or so. So it's mostly BSL 1. So that means you're just having a goggles when you're handling those. Mostly chemical lab, engineering lab, they use BSL 1 just to protect your body. And when the organisms you're handling with, so then it starts from BSL 2 and 3 and 4. And as you see from this video, 4 is the extreme. And I worked for the last 3 years on BSL 4, for the more than 10 years on BSL 3, and BSL 2 starting from my undergrad, postgraduate, like master degree. So BSL 4 is very important, and very few labs. I don't think India has. Oh, India has one lab which is in. No, I think it's in Bhopal. Bhopal, they have a BSL-4 lab. Gwalior, they're making BSL-4. I'm in contact with them. They have need my expertise how they are, organized those BSL-4. I don't know. The Pune has National Institute of Virology. Yeah, but I know they have the BSL-3 labs, but I'm not sure about 4. But that's in the United States. There are only I think 5 labs they have. The reason is the BSL-4 lab is very expensive to maintain because it's a lot of regulations. It should be also investigated by the foreign countries like CDC, WHO, FDA. They should come and visit. Your lab is equally compatible with the international standard. And also maintaining the lab, it should be totally sealed. There are no such windows or anything. The lab should be completely sealed so that the organism, if there is a leakage or anything, and it will be in a positive pressure, so air is coming in all the time. It's a high positive pressure, the whole room, so you cannot breathe there, so that's why you have to wear the suit, which is connected with the oxygens, and you breathe inside the suit. And when you go in, you don't need to shower, but definitely when you come out, you need a chemical shower, then water. So chemical shower means it is they call microchem, which is a chemical which can kill most of the pathogens, like 99.99%. You take 5-minute shower and then take a water shower, and then come out. Then that means the outer coat, get decontaminated. But inside also you are wearing your lab dresses. So you go to take out your lab dress, and take your own shower, and then you have to clean your body, your hair, everything properly, thoroughly, and then you can come out. And every exit and entry is well recorded. And for our case, because I was working in National Institute of Health, every day, who is going, who is coming out, what work you are doing, everything should be well documented and to be reported to the CDC for evaluation. If there is something happens in future, some outbreak happen, so they can track down who is working at what time. And all the labs has cameras, people can watch from outside. If somebody fall down, if somebody has a heart attack or something, it will happen, right? And you have an anxiety wearing those, I don't know, I cannot get out. So those things, they take care of it. And so it's just for your info, eventually in future, if anybody explore that area, working on BSL-4 labs or something in this country or outside, so, that's kind of a glimpse of it. Oh, exactly. So once you wear the suit, you cannot go out. So normally they recommended, like, you can stay there like maximum 4 hours, because you cannot go for urination. You cannot go, oh, I have to go to the restroom. No. Yeah, you cannot drink either. People, so sometimes I work there maybe more than 6 hours. I can feel I'm getting dehydrated because and exhausted, because the suit itself is several kilograms and you have to wear it. And that is giving you protection from outside pathogens. So you have to wear those. Those kind of, and you have to go to multiple trainings for this, how to do it. And then you got certified that, okay, you can go to that lab. And you have to inform the CDC, you inform the USDA that this guy, Bapi, is going to that lab and working. So they will approve it. They will see, okay, you had the proper training and all those. Then you get a license from the CDC and USDA that you can work for another 2 years. Then after 2 years, you have to renew it. Not only that's the good thing or bad thing, whatever the way you think about, but also you have to go through your physical exam. That means for how much cholesterol level you have. Do you have a blood sugar level? So those things they measure. Like, you have to be physically fit, basically. And I cannot have, like, obesity or overweight, and I'm going there. It's a kind of no situation. So you need to be properly physically fit. Your blood pressure is good, because I have to every couple of months they want to see your blood pressure. You have to say your blood report, your cholesterol level, everything they check out. And if they think it's little deviations, they recommend, okay, do something, make to that baseline what they want to. So yeah, so one way you be physically active too, by pressure. But it's also important, like when you are working there, you taking care of yourself, you're not getting exposed to those. Another thing I like to mention, like, especially for United States, like, and they cover the whole insurance treatment. Like, suppose, mishap happens, right? You're working something and you have, though, sharp objects is not allowed in BSL-4, because you have thick gloves, both hands, and you are working in the lab. But mishap happens, like you tear the glass somehow, or you put somewhere, open the door, or between doors, your gloves stick, and there's a hole. Or you by accidentally you drop something, you're carrying the flask, or 96 well plates or something. So you got exposed to that virus, because those are live viruses. In that case, or you cut yourself something. So then the CDC and USDA, they covered your, all expenses for the treatment. They have, Washington DC, they have special hospitals, especially for those who are working on BSL-4 if they have exposed to. So they'll take you there with a convoy, like President goes with in the front, the convoy is there. So they'll take you in there, not a good way, but a bad way, that you are infected. So that, and they'll take you in the hospital, they'll keep you there, and they'll take care of you. All the treatments is covered, because they're working for the government and also for the country. So that's good or bad, the way you think. But this is I also, it's a good experience. You have those. So. BSL-3 is not that rigorous, but you have to follow the safety protocols. They'll not take you by, all the hospitals are there. So this, they are, because BSL-3 is mostly like COVID, you can use BSL-3. Tuberculosis is BSL-3. Some of the viruses, today I'm mostly, whatever virus we'll talk today is mostly BSL-3 viruses. Again, BSL-3, there are some vaccines there, but not fully proven vaccines in the market yet. So that kind of viruses, and also there is a more chance there is pandemic or epidemic may happen. That's not, when BSL-3 it says, means it's not well known, okay? And it causes, a lot of infections and pathogenesis, and, may cause death too. So one of them is chanidpura virus. From the name it says it's India born, right? Chanidpura. It's again mononegavirals and rhabdoviridae. It's a vesicular virus, and its genome is negative single stranded RNA. It's a bullet separate virus because it's the same family where the rabies virus is. And the more important is this guy with the sandfly, which is actually transmitting the disease to the human. So here is a structure showing the Indian map, where you can see those color coded. This is confirmed case, which is a dark, I should say dark yellow color, and the light yellow is not reported yet, no report yet. But you can see majority of in India this virus is present. Okay. And what that virus does, so this is some of the glimpse of the characteristics of this virus and transmitted by sandflies. You can see the human exposure through incidental sylvatic cycles. We talk about sylvatic cycles and urban cycles. So that is also present here. Both sexes feed on plant sap, nectars, and honeydew, and blood meals are essential for female to give the eggs. So definitely, they are sandflies. They feed on those plants, sap or nectars, but they are the transmitting the viruses when they, ingest some humans, and they have the viruses, and then transmit to another when they bite another person again. They are nocturnal. They center in dark environments during the day. So what happens, a lot of, you can see the houses, they are not plastered, like, this is the plaster, right? The people have houses, they are not plastered. So what happened if you don't plaster, what happened, the humidity accumulates between the bricks, right? Because there is a gap. And those sandflies lives there and hide there inside. And those are the good places because the humidity is perfect, temperature is perfect, and they can hide there. And that's why they say, unplastered house is not good some way. This is one of the reasons, because the sandfly is there and they can be, transmitting those viruses to you. And other things you can read also, I cannot read from here. Scientists this is how long they are, and then I can see what is environmental factors, which is I'm saying here, like temperature, humidity, and latitudes. So those are important, because Indian environment is very suitable for them, because nice humidity, because you see both sides we have the sea, right? So when both sides covered with the sea, then a lot of humidity is there, and temperature is always there. It's not like if you go to Kashmir, there may snow, but most of them is like very, 40 to 30, that range always there. So they are good for their breeding. So this is a simple table which he got from how the outbreak happens. It started in 1965, but there is no cases reported. But then you can see there are a lot of cases reported till 2024, 245. That is the latest happened in Gujarat and Rajasthan, and 82 were died. So it's a high mortality, like 33%. The interesting, you should see the age group. You see, all are below 15 years. So it's mostly affecting the young populations. 15 years, like school, college, school level, that's kids, they are mostly affected. They have not seen anybody when the adults are getting infected. So what happens to those sandflies? So there are two kind of families are there. Phlebotomous vectors. This is one group of sandflies, and another is Serigontomias vectors. So there are two kind of sandflies are actually responsible for transmitting this disease, and you can see the distribution. And both the species are present in India. India has more the Phlebotomous, but the Sargentoomia is also present in Africa and also India and some Pakistan and all those Arab countries. And here also the same way it is present because you can see this is all the sea area and there is a lot of humid and all those. So why this is not happening? It's not the temperature. Like America is always cooler. Maybe the southern part is little hot, but they are unable to find those sandflies are present there. So again, like dengue or West Nile, we have discussed like it crossed the blood-brain barrier. So this infected sandfly when infect those humans, those viruses go to the brain, so they damage the blood-brain barrier and they get in. And so there, one method is the like Trojan methods, where the viruses by the Trojan methods they enter in the blood-brain barrier, or another is the direct effect. So they bind to the blood-brain barrier and make a pores there and got in. Once it get in, it always accelerates those immune cells infiltration. So mostly the platelets, neutrophils, some of the cytokine releasing cells like the macrophage and neutrophils, those are going in, and then you can see neuroinflammations. So you see the encephalitis manifestations. So you can see those. This is the report. What I was saying, like in 2024, there are Gujarat, like a lot of cases and a lot of deaths happened. So what are the risk factors here? The pediatric population, as I said, like children less than 15 years, they are mostly, get infections and death happening in those populations. Prevalent in low altitude and semi-arid regions with average rainfalls. So humidity is very important to grow those sandfly. They can breed and grow their colonies. I mentioned that unplastered houses are highly susceptible. The moisture stay there in between the bricks, and they hide there those sandflies. Close human-animal interactions happens. That's another way because they also have the sylvatic life cycles. So people are going to the forest or something, or animals went to the forest and came to the urban area, and human is interacting. There are chances that you can get also from the animals and the virus, you can get infected. During monsoon seasons and post-monsoon season, there is also a report that this virus is there. So, a lot of organs it targets. So definitely gastrointestinal. You can see those symptoms like you are vomiting, diarrhea, abdominal pain, anorexia, constipations. Respiratory, you have breathing problems. Ophthalmology, you have pain during eye movements. Those things, cutaneous, vesicular eruptions during cells transit. So you have like eruptions, like what happens, like we saw like West Nile, you can see rashes. They also have some kind of rashes, those, and which may be little like fluid inside accumulated there. And also it causes sometimes hemorrhages and disseminated, which is another important thing in the next slide, I guess. Hemorrhage, disseminated intravascular coagulation. So it causes clotting in the body in different places. And encephalitis because it crossed the blood-brain barrier. So there's the laboratory diagnosis, how to diagnose those, how they come out, how many cases are positive or negative. So RT-PCR, we all know that reverse transcriptase polymerase chain reactions. But these are the proteins they target. So you can see those are the glycoproteins, the envelope proteins, phosphoproteins, nucleoproteins, which is inside, and they have a matrix protein. So the PCR has been generated against those 5 primers, and you can detect those. You can also do the cell culture isolations by virus, so you get some tissue or blood, you just put in the cell culture and see cellular changes, like which typically in biology term called cytopathic effect. Cytopathic, I'll cover it maybe day 5 or day 4. So what is cytopathic effect? So you can change the morphology of the cells changes compared to the control. We always need a control to compare with. IgM capture ELISA, I think yesterday we talked about a lot of, like every viral infection, there's IgM comes first. We can talk more if you have questions. What is IgM, IgA, IgG? These are the 3 major immunoglobulin in our body all the time presence. IgM is a kind of pentamers. IgG comes later, it's a monomer, and then IgI, it may be monomer or dimer. Ig is more present in the gut and secretions in saliva and all those. IgG is more, IgG, IgM present throughout the body, secretions and everywhere. So it has different functions. You can read the books and all those, but that is another capture ELISA. We'll talk about what is capture ELISA maybe day 5 or 4. So how to do those capture ELISA, this is kind of sensitivity assay. I think for all biology assays, this is those kind of assays are mostly done. Neutralizing antibody assays. So if you know that virus, that's another way to see, are you generating antibodies? So that's people are doing neutralizing antibody assay. You coat the cells or with the virus, infect with the virus, and then put the antibody there and see that can neutralize the virus. So you don't see cytopathic effect. So that's indirect way to see, okay, I have the neutralizing antibody. So if you have a neutralizing antibody, it's a good thing. Means you can protect subsequent infections. There are a lot of animal model has been tested, mice, adults, everything. Indirect immunofluorescence, and there's another assays. You can look over in the tissue level. You can get a biopsy and see, okay, there's any changes, virus-specific pathogens. Like, if I do indirect immunofluorescence for glycoproteins, I have antibody tagged with glycoprotein tagged with antibody, and I have a tissue, like, I take a biopsy from some organs, like, I don't know what is happening, and then do that assay, and you can see signals of the fluorescent under microscope, and then you can say, okay, that is called indirect immunofluorescence assay. And also transmission electron, it's not normally used as a diagnostics, but it's like a gold standard. Like, I can see the virus because you need electron microscopy to see the virus because it's very small. So that's kind of diagnostic confirmatory assay that, okay, but you need special laboratory for this. In the electron microscopy, the technicians should be specialized how to do it, and you have a good facility to keep those electron microscopy and interpret the data. So it needs special skills to do this, and not every lab can handle it. Go ahead. Oh yeah, because there are so many different. Maybe I'll talk again day 5, 4. So they are like scanning electron microscopy, and so so it is a electron microscopy. You can read books. There are a lot of YouTubes there. So you can take a tissue, make a section. It should be cryo sections, okay? Cryo sections, and then you have the probe. So it's mostly like a gold level probe. And then you put it on the tissue. Like, I have a gold level probe against the glycoprotein, especially for chanidpura virus. And I put it on the tissue and incubate it, and then see under microscope. And if I see, I see some golds there. And then you can find out, okay, because it's binding to the viruses directly, and then I can detect the virus directly, and you can see the structure and everything. Sometimes I'll show another slides. Influenza, I'll show you that how the virus looks. Then you can completely see the virus itself. Now, how the virus looks like, and that's only can be visible by transmission electron microscopy. So that's only you can see viruses, because otherwise normal microscope cannot capture those data. It has no resolutions because transmission has a high resolution. You need 40,000, 50,000 resolutions to see those small microorganisms. Management is more important here. The no specific antiviral drugs, no vaccines. That's why it's a BSL-3 still, and it's very prevalent in India. So always it's kind of a you should be proactive. You should make sure that if you're staying in a hostel or in a dorm or any places, make sure there's no supplies there. But the age is everybody crossed already 17 or 15 years. But for the young kids, staying with you or somebody, for them they are very susceptible to those. So those are very important to take care of it. Yeah. I don't know. I don't know. Because this is mostly localized in India, and I think Indian scientists should do some research because USA we cannot get those viruses until we collaborate together. If you guys interested to collaborate, we'll be happy to collaborate with this because we cannot get the virus. Because we need, yeah, we have the facility BSL-3, we can do all kind of things, but we need to get the virus. That needs collaborations. And then if we do collaboration, we can definitely get a very good paper, very good studies, why it is affecting. That's very good questions. We can generate good models and why, very young age. What is the difference? Definitely immune function is more important, but 15 years, they have already matured immune cells already there. So why they are targeting young kids, not the adults? So I don't know. I have no clue. But I think it needs more study there.
Maybe as no vaccines available for kids, as I was mentioning, COVID did not affect the young kids.
Exactly. COVID is different, right? Yeah, COVID is different. Like COVID, how many deaths happen? Maybe very few. Maybe exactly opposite. Exactly. So exactly. So we don't know what Chandipura does to young kids, why they are more susceptible, or they are getting more exposure, more virus load. Somehow maybe they are standing like in the wall where so many bites happening, right, in the backside. But the adults may not doing those. Maybe the kids are playing together in an unplastered house or somewhere, or going to the forest, or get more exposure than the adults does. I don't know. Those are kind of hypothetical thinking, but it needs more elaborate, kind of very precise experimental design, and then do those studies. I think that will give some more answers on those. Yeah.
Vaccine is not available. Is it difficult? This inactivated vaccine can.
So yeah, so there are some preclinical vaccines there. So preclinical means it's not tested yet in the humans. It's mostly in the animal model they tested, but the efficacy is not that great. I mean, so far they found in mice model, but mice human is a lot of different immune functions. Mice macrophage is totally different from the human. Innate function is totally different from human. So it needs more study. Theoretically, it needs more study. But there are a lot of ways you can make vaccines.
Especially the renal one, they get affected because pharma industry they have to come out with vaccine. Then always they look at taking profit also.
Exactly. So there is another thing, right? Yeah.
Only in case is
They are calculating money, right? Yeah. So I mean, I think it's the government should come out with funding. They're definitely DOD, Department of Defense, Indian government, or ICMR, by the DBT. They should come up with that kind of proposals that hey, we are funding for Chandipura virus making vaccines, and give a more effort for the next 5 years and make something. Right, then maybe institute maybe interested to, oh, I have gotten funding, so let's do something. So that kind of initiative should come from the government side, or maybe somebody private. I mean, he's right, like why private will spend when only few are dead there, and so it's only maybe sample, but but I think it's very important because you can see it's very few states are not touched yet, but maybe not reported yet. But maybe they are, but it's unreported. So many things happen, right? Unreported cases.
I'm having clearing it for those.
Exactly. So exactly. You're staying in Bangalore, you don't grow. So what about the rural area, right? So that's the thing here. So I think that's the initiative have to be taken. definitely from the Ministry of Public Health and those sectors. And then they should provide more funding to the institute that, hey, you have a facility, just do it. Yes, and it needs BSL-3 facility so they can do better research there and do it, because India, there are a lot of BSL-3 labs there, so they can easily handle it. Yeah. Repurpose, drug repurpose, this terminology may be new to you, but we'll talk again for 5, day for 5. So it's kind of touch on that. What? Yeah, ribavirin is mostly hepatitis. The flavipiravir is also, I think they use for hepatitis and some of the hepatitis C. So what happened, repurposing means I have a drug which is specifically used for this virus, like Tamiflu. Probably you heard, like if I have a flu and a severe flu, the doctor first recommend, oh, take the Tamiflu immediately. Okay, so I take the Tamiflu. It never happened to me. My daughter happened. So take Tamiflu. Tamiflu is specifically designed for flu. But if you use that drugs for another virus, for example, chanidpura, I like to test. Oh, it works, right? Then it is repurposing. So my same drug I can use multiple purposes. So it's a kind of same. That's what it says. Repurpose, right? That we are using for another purpose. So that's the drug repurpose. Right. Yeah, even like COVID, right? So many people use that ribavirin, and so many drugs people have used, corticosteroids. People try those. It works sometimes, some people, sometimes not. It's like a 50-50 chance. Right? So those are the vector control is a major thing, and PPE against other vectors, dress well so that take repellents on those things. Make sure you're not some places where there are more chances of sunlight, those kind of things. So it's kind of more awareness things here. Okay, this is done for Chandipura. So you learned something, right, in Chandipura, I guess. So next maybe we can start another one, Dr. Reddy. We can start another. Huh? Oh, break for coffee. Okay. We'll take a coffee break. You can too.
No. We'll wait. If it's ready, then we'll go for coffee break.
Oh, okay. Then otherwise we can look the Nipah virus. Nipah. Yeah. Hanta, yeah, that's right. So another thing I'd like to talk, the hanta virus. I mean, it's also prevalent in India. It's another BSL-3 pathogens. And as you know, I'm just repeating again because BSL-3, there's no vaccines, drugs are limited, that kind of thing, right? And it's contagious too. And so this is orthohantavirus. It's a Hantaviridae family. In Bunia virus is the order. So order is the first, like multiple, like a C, and then its family is smaller, genus smaller, and then species more defined. Okay. So this is the virus. It's a schematic one. Okay. So you are saying how the electron microscopy pictures. This is so once you do electron microscope, you can see the whole virus, how the virus looks like. It looks like very hazy, but it's electron microscope, the pictures is coming like that. Yeah, that's why electron microscopy is it's a gold standard, and two gold standard. One is transmission electron microscopy, another is a virus culture. By culturing, you can understand, and then you can do more study on what viruses. You can purify those viruses. You can look over the proteins and all those. So those two are the major gold standard. The others are kind of indirect methods, right? I'm measuring IgM to see, okay, it has a Chandipura or hantavirus or not. So all those are indirect methods. RT-PCR is another, but it needs specializations, The PCR, you make sure it is not contaminated. Your lab is not contaminated. There's so many RNA, DNA in your lab. You are handling properly. The person who is handling, and he has a proper control, positive control, negative control. If you quantify, just like real-time PCR, if you do cyber green or so, you have a nice car, you have to show, okay? Okay, I have a nice car I got by cyber green, real time, and here is my uh, GC value, right? Okay, how much copy, because they are you are measuring the copy number of viral copies. That's why people do how much viral load. They measure the copy numbers. RT-PCR is kind of yes or no questions, right? But if you want to see how much virus you have present in your body, then you have to do those kind of quantification, which is real-time PCR. We can talk more day 5, 4. So this is the virus, but the interesting part of it, it has a 3 different segments. Okay, one is big segment, so it's a segmented RNA. It has a one is large segment, one is medium, and so small. And there is also each segment they have like RD, so RNA dependent RNA polymers, which is very important for making copies. Okay. And they are trimeric N proteins. The N proteins are always present inside, and the outside it has a glycoproteins N and glycoprotein C. So they are in the outside. So it is a glycoprotein, which is a major thing of giving protections. If you generate antibody, you should generate against the glycoproteins to give you protections. There are a lot of viruses. I think this time the slide is very much better, so I fixed it. So there are a lot of viruses you can see. The Americas, that means North America, South America, how many viruses are there? And Europe and Asia. We are more focusing on Europe and Asia. And you can see the Hantan virus, Kumala virus, Dobrava virus, it's mostly Russia. Seoul, Tula, we just have accepted one review article in one journal. I will share it. We talk about all those recently. So that paper has been accepted. So these are the common viruses, okay? And you can see the syndrome. HCPS. So hanta virus, here is a HCPS, hemorrhagic fever, renal syndrome, and HCPS, hemorrhagic hantavirus cardiopulmonary syndrome. From the name it says, when it says HCPS, so it affects the lung, okay? And when it is a renal R, like this, HFRS, it's mostly in the kidney, okay? So though it came from the same virus, it has a different targets. One goes to the lung, another goes to the kidney. We don't know why. So, but it's all the same family. So that is a very characteristic of this virus. So this is the first virus I think nobody has heard about. This has been first detected in Vellore, 1964. There's first suspected cases. They named on the regional virus name like Protopalayam virus, and later it's called protein virus. Okay, it has been named as the regional virus, and it has been 1964. 1998, 1999, there are a lot of antibodies. So they got seroconversion. So they find out antibodies in the patients, but they cannot detect the virus. But you see the 1998, 99, maybe the technology is not sophisticated enough to look the viruses. They just got the blood, plasma, and detect some antibody, and that's it. So they found some antibodies, which is same like the Kumula virus. Before 2000, no clinical documented hantavirus. Again, maybe there is this, but it's not reported. Who knows, right? That early. 2000, there are 2 fatal cases reported, one in Chennai and one in Cochin, from that viruses. But it's very sporadic, right? You see like 1 case, 2 case, but again, it is the start, right? When there's like how the COVID happened, right? It's the start. It happened in China, and then it fell up everywhere. Maybe because the COVID is very transmissible virus. This is not that transmissible, but it has a potential to do big things, right? So that's why I put in this, lectures that this is another important virus, hantavirus, which is prevalent in India, and we need to do more research here, or maybe some collaboration with others in countries or so, to understand more how to control it and those things. So this is another presentation like between 2000 to 2020. It's mostly prevalent. India has not been because it's so sporadic, as I've shown, like one or two cases like that. But it's highly prevalent in China and Europe and Russia. So definitely, people are traveling from different countries. May happen like they got the virus from other countries because they are all in Asia, right? Except that this is Europe, but Russia and China. So there is possibility that the virus came from there, or who knows. And so the North America, this is separate because they are mostly affecting in the lung. This is most affecting in the kidney. So the same like virus replication, always it's the same like what we discussed yesterday. It has a receptor, it goes in, then here like transport vesicles endocytosis happens, and then as I mentioned, just recapitulate yesterday, like endoplasmic reticulum, where the non-translational protein, non-structural protein synthesis happens. Okay, and the nucleus mostly the replications happens. If it's negative sense RNA, it goes there. Positive strand happens, which is called complementary RNA, and from complementary against other RNA forms. And the Golgi is the trans and cis. So Golgi is like a packaging station. So I got nucleic acid from nucleus. I got proteins from the endoplasmic reticulum, and they are packaging the viruses here. And after packaging, they release. Okay, that's the idea for all majority of the viruses. They do this same principles for replication. So again, it is transmitted by the rats. See, so the rats are the most, and there are two different species of rats. So this species is also prevalent in India and other like China's, and this is mostly the Americas, oligochomous species and like apodermos species. But their interaction in the humans after transmissions is different. Mostly the Asians, they are affecting in the kidneys, and that's why it's a renal syndrome. And others, they are mostly affecting in the lung and heart. It's more pulmonary cardiac syndromes that happen on that side. Okay, so like here in the example, this Andes virus, which is very common in Europe, and here is mostly hanta virus, which is also prevalent in India. So there's overlapping signs. Show the next slide. The overlapping signs is like this. The lack of platelets, proteinuria. Through urine, the protein is going out. That is common for both syndromes. The laboratory test, if you see, it's not clear here. Right? Renal syndrome and cardiopulmonary syndrome, both are same, like a loss of platelet thrombocytes. Both cases, loss of proteins from the urine, increase of creatinine, hematocrit, all those things happen. But clinically, the pulmonary affect the respiratory system, and there is also cardiac, so there is hypotension and respiratory failure. And whereas the renal, it is mostly kidney failure or hypotension. So that is the difference here. And we can see here the most of the symptoms like fever, myalgia, headache, those things are common. I think you have seen for all the viruses, right? It's very generic, like those fever is always, because why fever happens, it's the same thing, like COVID fever happens, HIV fever happens, this virus fever happens. It's all mediated by the cytokine chemokines, which is called pro-inflammatory cytokines. So what are those? IL-6, TNF alpha, IL-1 beta. So those are the major causing those things. Fever, myalgia, headache, those things. Those, if the cytokine increase, that means you'll get it. And all viruses do those pro-inflammatory cytokines increase, and that's some common symptoms it happens there. So this is interesting. So maybe spend some time here. So what happened to the so this is like normal endothelium, okay? So this is the blood vessels. And this is like all the cells in our body, like bind together. So in between those, these are the tight, we call tight junction protein, which is like a VE cadherin molecule. We have shown in the gut a lot of papers, others did. So it's the cadherin molecules which binds those cells together. So there is no leak. When we have this protein breaks, there is a leak. So when infection happens, so there is a gap. You can see there is no gap. There is a gap. So what happens that that VEGF, so VEGF is another chemokine. When it is upregulates, it breaks those cadherin molecules, so there is more gap between cells. And when there is a gap, so then there is a porosity, and then the cells go out. So that's why hemorrhage happens. So from blood cells, red blood cells go out. So many things going out because the virus is inside the cell, and also because there is a gap. The platelets and neutrophil, they are the cells which comes first. Oh, there is a gap. I have to do something. So they come and they try to make it block. Okay, so they come together. Like when we have a cut somewhere, we see it's oozing, then it suddenly stop, right? Or you press it, then it stop easily. Why it happened? Platelets came in. Platelets are very sticky cells. They came in. So same thing like here, the platelets came in because they saw, oh, there is a gap. Blood is oozing. I have to do something. So they make a lot of platelets came in, and they finally make it clot. So you can see there is a clot. So what happens with this disease? You generate a lot of clots in your body inside, which is not good. So the coagulation is happening, and then the complements always come. And then bradykinin is also upregulated. Bradykinin also helps to break those permeability. So that happens in this case. So here, for example, in cardiopulmonary, so this is the structure of a lung, and different views. So basically, focus on those. Like acute phase, you can see it's very, very like an opaque. It's not clear enough. So here it is clear enough, but the acute phase and weaning off mechanical. So because they are giving ventilations, because the person cannot breathe well because it affected the lung. So it's all it's full of exudates and all the fluids, so they cannot breathe well. So that's why you need intubations, give the tube so that through tube you are breathing. So basically their opacity is there in the chest X-ray, and that's the characteristics of this disease. So what they do, there's only supportive therapy. There is no specific antiviral therapy, no immunomodulatory drugs are present. So only the management strategy is careful monitor clinical signs, give some fluidics, electrolytes balance, blood pressure, maintain blood pressure with some medications, try to maintain your blood pressure, urine production because it affects kidney, so make sure you're drinking enough. All are symptomatic, like analgesic drugs and all this. The major important I like to talk about ECMO. So what happens? There is a machine. You can do those things. Like you put a machine here, and you collect the blood. We did an experiment when I was in Chile with monkeys. So we collect the blood from the veins. Okay, collect the blood from veins. This is a venous blood. And then you put in the machine. So in the machine, it filters the blood and also put the oxygens in the blood. So you get the more oxygens in the blood, and then so there is a filter. You're filtering this blood and then putting oxygens too much so that the blood get oxygenated, and then put it back to the human body. So basically, you have a catheter through the vein. There is a machine there, it goes and circulating back to your body. So you are putting oxygen. Basically, you're giving oxygen and purify the blood, because if there is any impurities, any clot. Basically, you're generating clot, right? You should not get a clot and die due to stroke or something. So that's why it's the preferred method, like collect those bloods, oxygenate it, put it back. So that is another therapy. It's mostly used for this disease. We tried for people gave a company approached me and say we are doing the HIV research that time, and say, hey, we like to use the same ECMO protocol for HIV. I said it should not work. The board said no, because they are paying money. I thought, okay, let's do it. So they did it. It does not help because the virus, it can purify some of the viruses, right? Because it's filtering that. It take out some of the viruses. But you have to make some kind of adjustment, because if you filter too much, you're also filtering cells. Cells are bigger than virus, right? So you are not, so you have to compromise there. So what they did, they put some radiation, UV radiation. So we put some UV radiation at that point, so that the virus got inactivated, and then put back those blood to the vein, the venous blood. Okay, yeah, venous blood to the artery. So the idea was to somehow by UV radiation kill those HIV viruses and put back those, so that the monkey will be cured from the HIV. But no, because HIV is totally different funda, because the viral reservoir is already in the lymph node, and then when they say, oh, there is no virus, the lymph node try to produce more viruses, and it circulates again. The experiment failed actually. We are unable to do anything. But then again, that company approached me can you do for COVID? I said no, because COVID is also has some reservoirs. It goes to the brain. You probably heard a lot of papers came out. It causes brain hemorrhage and others. So those and even lung, it resides there too. So it is also difficult for COVID to completely eliminate. They have not, reported anything that COVID has a reservoir by itself, but there are some viruses always there. So we never approach that. But yeah, that's kind of information for hantavirus. But again, I think nobody have heard about, right? You have an outbreak in Cochin, you have an outbreak in Chennai, but it's sporadic. But another thing you have to remember that that is another re-emerging disease, and it's BSL3, and I think more research to be done to understand those pathogenesis of this virus better. So.
Any questions? We're ready for talking.